Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled 10-Minute Charge Heterogeneity Analysis for the Development and Quality Control of Biopharmaceuticals. Two of the most critical steps in protein characterization and biotherapeutic development involve the determination of protein isoelectric points and charge isoforms. These factors play key roles in biological activity and drug stability. Thus, they can also be considered potential make or break points on a product's path to commercialization. Today's webinar has been designed to help you carry out these analyses more efficiently, effectively, and accurately. Let's meet our expert panel. Dr. Scott Mack is Senior Scientist at ProteinSimple. Scott will discuss the development of a robust charge heterogeneity assay for the characterization and quality control of biotherapeutics. Carrie Anderson is Senior Scientist in Vaccine Analytical Development at Merck Co. Carrie will talk about application of imaged capillary isoelectric focusing, or ICIEF, in vaccine and biopharmaceutical protein-based products. Karan Shah is Senior Research Associate 3 in Analytical and Pharmaceutical Sciences at Immunogen. Karan will describe the analysis of maytansinoid antibody-drug conjugates by ICIEF. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So, if everybody's ready, let's get going. Scott Mack will be our lead-off speaker. Scott?
Thank you very much for the introduction, John. So I'm going to talk about charge heterogeneity analysis by ICE today. Charge heterogeneity is a key attribute used to assess the stability and capability of a biological manufacturing process. Many of the critical posttranslational or chemical modifications that can occur in a protein structure will affect the ionization of that molecule and will be captured by a charge heterogeneity analysis. As compounds become more sophisticated and characterization of the production process becomes more demanding as those occurring in QBD efforts, analytical chemists face unique challenges. They need to generate better data faster in order to help speed up and improve product development. Analytical tools need to keep up with the demands placed on their operators. They need to be faster to generate timely data, robust to reduce efforts generating that data, highly resolved in order to detect minute differences between preparations, and flexible enough to support general separation techniques. And in the case of IEF, they provide additional identification information such as isoelectric point. When comparing charge heterogeneity techniques, it becomes obvious that the ICE system meets these demands better than any other analytical system. In 10 minutes, a highly resolved or reproducible charge heterogeneity profile can be separated. This represents a 5 to 6-fold improvement over traditional CIEF or ion exchange method. Capillary isoelectric focusing, in my opinion, is the most powerful separation technique in all of CE. For those new to CIEF, this mode of separation has some distinctive features that are important to understand. The first step in any separation is sample introduction. In CIEF, the entire capillary is loaded with a sample solution containing PI standards, ampholyte, methylcellulose, and protein. One end of the capillary is immersed in an acidic solution and the other in a basic solution. Once the sample is loaded, the sample is focused by applying an electrical potential across the capillary, which drives the countermigration of hydronium and hydroxide ions into the capillary, titrating all amphoteric compounds to their zero net charge, known as their isoelectric point. The best solution for CIEF detection is to take an image in the entire capillary. Eliminating the mobilization step dramatically accelerates the CIEF separation process. It also avoids distorting the delicate pH gradient through the mobilization step. The entire gradient can be observed, providing critical information on the focusing gradient, which greatly simplifies method development and troubleshooting. Imaging during the entire CIEF separation, the ICE system allows us to capture the complex bidirectional migration pattern of the sample peaks as they titrate down to their isoelectric point. The ICE's ability to monitor the forming pH gradient during focusing provides tremendous operational advantage over traditional CE systems. And here we can see actually the formation of merged peaks from the anodic and cathodic front. And eventually you come with formed peaks. The ICE system has a very elegant analytical workflow. Between each analysis, the capillary is cleaned with a rinse of methyl cellulose. The rinse time is usually 1 minute or less. The sample carryover is not as severe in CE as LC. Following cleaning, the capillary is loaded with sample in a second 1-minute rinse. Once the capillary is loaded, the sample and the amphoteric components are focused with the application of an electrical potential for a typical range of 6 to 10 minutes. During focusing, the images are taken of the capillary every 30 seconds, with a final image taken at the end of the focusing period. CIEF presents a unique challenge to traditional CE systems, which are designed to handle moving peaks. Nearly stationary, the focus bands require a mobilization technique for peak detection. As you will see, the mobilization techniques, though effective, are not without their disadvantages. Now, applying pressure to the focus gradient in a traditional CE system can allow for detection of the analyte components, but this method, while preserving the spacing of the gradient, leaving calculated PI unmolested, the hydrodynamic force used in this method introduces laminar flow in the capillary, significantly reducing resolution. Resolution can be maintained through the chemical mobilization process. Typically, replacing the catholyte solution with a weak acid or salt reduces the pH of the gradient, adjusting the pH gradient past the detection window. The major disadvantage of this method is that it disrupts the relative spacing of the gradient, which can create large deviations between actual and calculated PI, especially in the range of pH 8 to 9. As published by Pfizer, ICE has been found to be a highly flexible separation technique capable of analyzing entire pipelines with one set of conditions. The capability to support platform-based methods has the added benefit to allow analysts the ability to leverage prior method development efforts in the analysis of new compounds. Beyond longer separation times, high surface area to volume ratios can lead to mixed mode separations that significantly limit the separation capacity of an ion exchange method, typically for a larger compound. This not only limits the resolution, but also in many cases requires molecule-specific methods to generate useful charge heterogeneity data. This side-by-side comparison, and I do want to take note that actually if you can switch the labels from ICE to ion exchange, they're actually switched. So the higher resolution separation is the ICE system. It's important to take note that unlike surface interaction-based separation of chromatography, the electrical field of CE can also penetrate into the three-dimensional structure of a protein, interrogating differences within the solvated pocket. Highlighting the robustness of the ICE, here's a run set recently generated during the validation of a new cartridge type. As you can see, the charge heterogeneity profile is stable in both terms of PI and peak profile for at least 100 runs. In summary, when faced with challenges of charge heterogeneity analysis, turn to capillary isoelectric focusing by ICE. You will not be disappointed. It is the most direct path to generating rapid and robust high-resolution charge heterogeneity profiles across a wide range of protein classes. Now back to you, John.
Scott, thank you for that detailed overview of the use of ICE to determine protein charge heterogeneity. I think you've really covered a number of important topics and helped set the stage for the presentations to follow. If you're just now joining our webinar, welcome. As I mentioned earlier, we'll be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Carrie Anderson, will now begin her presentation. Carrie?
Thanks, John. Today I'll be speaking a little bit about some of the applications of image capillary isoelectric focusing in vaccine and biopharmaceutical product development. In the past 2 decades or so, capillary electrophoresis has grown as a powerful separation technique with high separation efficiency and low sample consumption. In addition to the advantages of low sample consumption and its powerful separation characteristics, compared to the traditional slab gel techniques, CE also has advantages such as speed and the ability to quantitate and the ability to automate. And for these reasons, many are working to develop CE methods to either complement or to replace the more traditional, labor-intensive and time-intensive slab gel techniques. There are various analytical techniques which are available to characterize charge heterogeneity, and I've highlighted some of the most commonly used ones here in this slide. Each of these methods has its own advantages and disadvantages. For example, if you think about ion exchange and IEF, you notice that a sample injected onto an ion exchange column can easily be fraction collected, but there are cases where sometimes IEX lacks the ability to provide good separation of charge variants. Whereas CIEF has much better resolution than IEX, separated peaks cannot be collected. And for these reasons, we often use multiple methods to fully characterize the charge heterogeneity of our protein of interest. But because of its high resolving power, rapid method development times, and quick run times, image capillary isoelectric focusing is a very powerful tool, and it is suitable for first-line bioanalytical analysis and can be used for a variety of applications in product development. In my talk here today, I will be focusing on how we have applied image capillary isoelectric focusing technology in biopharmaceutical development, and specifically I will be showing some examples of how we have developed methods to confirm product identity, to evaluate product stability with respect to charge heterogeneity, to support clone selection and formulation development, and also how we are able to use an IEF method to determine concentration in process samples, and also to evaluate antibody-antigen binding. So I'll start off by talking a bit about product identity. Biological product development regulations stipulate that product identity testing is required for a site that produces multiple different products, and that the identity test should be highly specific and be based on unique aspects of the product's particular protein molecular structure. And traditionally, ELISA is often used as an identity test for biological products. This technique is usually time and labor intensive and often relies on several critical reagents. Another method commonly used for identity testing is peptide mapping using reverse phase HPLC following digestion with trypsin. Although this method may be more robust and is usually quicker than ELISA, it can still be somewhat time-intensive owing to the enzyme treatment. Shown here in this figure, you can see the electropherograms of 7 different monoclonal antibodies and 2 therapeutic proteins. And you can see that each product has a unique PI, charge heterogeneity profile. And I think it's important to note that in the case of monoclonal antibodies specifically, there is the potential for some monoclonal antibodies to have close PI values for the peak. You can see that here in this figure if you take a look at the electropherograms for monoclonal antibody 1 and monoclonal antibody 3. But you'll notice that even though the PIs of the main peaks are similar, you can see that the charge profiles are distinct. So by comparing the PI value and the IEF profile, we can use these two in combination to confirm product identity. And so I think it's important to note that image capillary isoelectric focusing is an ideal method for product identification because it's a relatively fast experiment, typically requires no sample pretreatment, and is more robust compared to the traditional labor-intensive techniques such as ELISA and trypsin digest reverse phase HPLC. And for all these reasons, the method is easy to run routinely and easy to implement in a QC environment. Okay, so now I'm going to switch gears a bit and talk about how we have used image capillary isoelectric focusing methods to monitor changes in charge heterogeneity over time during product storage. This figure here shows the electropherograms for a monoclonal antibody stored at 4 different temperatures for 6 months. And although I don't show the data here, I would like to take a moment to point out that the basic peaks for this antibody were confirmed to be the C-terminal 1 and 2 lysine variants by treatment with carboxypeptidase B. If you take a look at the left here, you can see that at higher temperatures, the acidic peaks clearly increase. And we believe these two acidic peaks are likely due to a chemical modification, such as deamidation, since we did not see any evidence of either clipping or aggregation at higher temperatures. Deamidation occurs as a result of conversion of asparagine to aspartic acid or isoaspartic, and its rate of conversion is known to depend on several factors such as pH, temperature, the amino acid sequence, and protein structure. And this here shows the long-term stability of the same monoclonal antibody evaluated in two different formulation buffers. This figure here shows the percent of main, acidic, and basic peaks plotted against storage time. If you take a look at the figure on the left, which is the monoclonal antibody, the formulation buffer at a pH of 6.5, you can see that the acidic peaks are slowly increasing, while at the same time we're seeing a corresponding decrease in the main and basic peaks. However, if you take a look on the right, you see that in the same formulation buffer at a pH of 6.0, the percentage of acidic peaks do remain fairly constant. And again, these results also provided some evidence that the acidic variants that we were seeing for this monoclonal antibody were due to deamidation, since the rate of deamidation is known to be faster at higher pH. And now I'm just going to provide some information here on a similar study we did. In this case, this particular protein is a recombinant protein used in one of our vaccine candidates. And here you can see the electropherograms for this particular protein stored for 3 months at 2 to 8 degrees C. So again, if you take a look at the bottom at 0 months and then the top at 3 months, you can see that the acidic peaks have increased while we do see a decrease in the main peak over the 3-month storage period. We did also monitor this product stored frozen for 3 months and saw no change in the product profile when the material was stored frozen. Again, in this particular case, we did not observe any changes in aggregation. We did not see any clipping. We did not detect any changes in binding using our ELISA method over the 3-month storage period. And since no changes were detected in these other stability indicating assays, we believe that the two acidic peaks here are likely due to deamidation, particularly because the amino acid sequence of this protein does contain 2 deamidation hotspots in the receptor domain. And I think it's important to note here that in this case, the image ICIEF assay was instrumental in providing some information on product storage conditions. And as a result of this short stability study, the decision was made to store the product frozen. Now I'd just like to provide 2 examples of how we've used our ICIEF method to show interaction between antibody and antigen. And this figure here shows a titration of monoclonal antibody with its target protein. The electropherogram on the bottom is the monoclonal antibody by itself, and above you can see the results generated when we titrate in the antigen. And I do want to take a moment to point out that in this particular example, the target antigen protein contains no tryptophan and only a few tyrosine amino acids. We do not actually see an antigen signal in the electropherogram. We just see the monoclonal antibody. Monoclonal antibodies in general are known to contain charge heterogeneity, and the acidic forms of some monoclonal antibodies have been demonstrated to reduce the binding ability to their antigens and therefore lower potencies. Therefore, assessing the binding ability of these acidic forms is normally evaluated using a combination of different analytical methods and can be a very tedious process. So if you take a look at the figure here, you can see that the main peak of this monoclonal antibody shifts upon binding with its target protein from a PI of 9.2 to about a PI of 8.7. And in addition, all of the acidic peaks of this monoclonal antibody have also shifted together with the main peak, indicating that all of these acidic forms are active in terms of binding to antigen. And we also performed a similar binding study with our recombinant protein, the same recombinant protein that I discussed earlier. This figure shows the binding of the monoclonal antibody that targets this protein. And upon titrating the product with its specific monoclonal antibody, you'll notice that the electropherograms begin to change, and they do stop changing about a 1-to-1 mole ratio. And again, you'll notice that the acidic peaks of this protein have also shifted together with its main peak, again indicating that these acidic forms are active in terms of binding to the monoclonal antibody. And if you remember earlier when I discussed the 3-month stability study we did test the 3-month sample by ELISA, and we did not see any change in binding. And this agrees well with what we see here. So again, we were able to show rather quickly that the acidic variants for this protein are also able to bind to its monoclonal antibody. And in this figure here, this just shows the results obtained when the same protein is incubated with a nonspecific monoclonal antibody. So you can see here in red at the top is our protein of interest, and below that in blue is a nonspecific monoclonal antibody. And then the black on the bottom, you can see the electropherogram generated when the two are mixed together, and you notice that there is no change in the profile of either the protein or the monoclonal antibody. Now I'm going to spend a few minutes talking about how we were able to use an existing ICIEF method to determine concentration for processed products. We were asked to develop a method that was suitable for measuring product titer in both upstream and downstream process intermediate samples. Now this figure here just shows the electropherograms for 5 different concentration standards generating using our purified products. I'll be showing the electropherograms for the actual process sample shortly, but I wanted to take a minute to provide a little bit of background information about how we ended up using IEF to determine concentration. Initially, we attempted reverse phase HPLC, and we screened a number of different types of reverse phase columns. However, because of the strong binding affinity of this protein to the column, protein carryover was a major issue, and so we were not able to develop an HPLC method to determine concentration. At that point, we decided to evaluate whether the existing established image capillary isoelectric focusing method could also be used to determine titer to support process development studies. And so the first thing we evaluated was standard curve range and looked at reproducibility of the standards. And you can see this figure here again shows the electropherograms 5 concentration standards prepared using our bulk purified product. And from this data set, we were able to estimate LOD and LOQ and also assess linearity. So standards were prepared and measured on 3 separate days to evaluate reproducibility. And as you can see in this figure here, the calibration curves for all 3 days are linear over the range of 63 to 500 micrograms per mil and have correlation coefficients greater than or equal to 0.99. And if you take a look, you can also see that the percent RSC is less than 5% for each of the standards. This figure here shows the typical electropherogram for the types of samples that we needed to analyze. And during product development, titer yields need to be analyzed early in the process in upstream samples such as lysates, as well as after downstream purification steps. And this poses somewhat of a challenge analytically, since the crude lysate samples contain many other cellular proteins in addition to lipids, carbohydrates, and cell culture media components. And here, if you take a look, you can see the electropherograms of the process samples that we needed to analyze. Samples 1 and 2 on the bottom are the upstream cell lysate samples, and samples 3 and 4 are 2 downstream samples generated after different chromatographic processing steps. And in green on the top, you see the electropherogram of our final purified product. So if you take a look at the electropherograms for the 2 lysate samples, you can see that these samples are clearly impure and contain many other proteins. And again, if you look at the PI of the main peak, you see that it is slightly shifted from samples 3, 4, and our final product. And this is likely due to a matrix effect. And we did confirm that the product peak here, observed at a PI of approximately 5.8 for these samples, was our protein of interest by spiking in pure product. And in addition, we also did spike recovery studies to assess accuracy. Recovery was determined to be approximately 80% for sample 1, the dirtiest sample, and ranged from 90 to 110% for the other sample types. And because this method did not suffer from the protein carryover that we observed using the first phase HPLC method, we were able to utilize our existing capillary isoelectric focusing method to measure titer. So finally, we measured reproducibility by evaluating a set of samples over 3 days. If you take a look here, you can see that the percent RSC is highest for sample 1, which you would expect since it is the dirtiest sample, and we saw RSCs of less than or equal to 5% for all the remaining sample types. Okay. And finally, we are also able to measure concentration during clone selection studies. So in addition to be able to provide some information on the charge heterogeneity of the proteins produced by different clones, we're also able to measure concentration using this method. And then I'd just like to wrap up my talk by providing two examples of how we were able to use image capillary isoelectric focusing for strain selection for glycoproteins. This figure here shows the image capillary isoelectric focusing electropherogram of recombinant human erythropoietin produced from 3 different strains. And EPO contains 3 end-linked glycosylation sites that have glycans with terminal sialic acid. And if you remember, EPO in CHO cells contains a heterogeneous mixture of sialylated and glycosylation structures. These particular EPO products shown here were produced from a Pichia pastoris strain that was engineered to produce a more uniform, sialylated biantinary and glycosylation structure. So during selection of the strain producing the most uniform and glycosylation pattern in EPO, we were able to use image capillary isoelectric focusing to monitor the charge heterogeneity caused by differences in sialic acid distribution. In this particular sample, you can see that strain 3 has the least amount of sialic acid heterogeneity since it contains primarily the 2 main peaks when compared to strains 1 and 2. And this assay was particularly important during process development because we were able to quickly provide an answer on sialic acid distribution, most times in the same day that the sample was edited. And then in order to confirm that all the peaks that we saw in the electropherogram were due to differences in sialic acid distribution, we treated the protein with sialylase A, an enzyme that specifically removes sialic acid. An unsialylated EPO protein has a theoretical PI of 8.4, and you can see here in this figure that when we treat the protein with sialylase, that all the peaks between about PI of 4.5 and 5 disappear, and we see this new peak that forms at approximately PI of 8.5, indicating that the peaks that we see are due to differences in sialylase. And during biosimilar product development, internal candidates are often compared with innovator products. And this particular figure here shows the electropherograms of 2 in-house strains compared to an innovator product. The particular protein here contains 3 end-linked glycosylation sites and multiple O-linked glycosylation sites in which the terminal end of the oligosaccharide has many sialic acids. This sialic acid distribution makes this particular glycoprotein very complex in terms of charge heterogeneity, which you can clearly see here when you look at the electropherogram. If you take a look at the figure on the left, you can see the 2 in-house strains are still different compared to the innovator product, but that the sialic acid distribution of strain 2, shown here in the middle, is slowly approaching that of the innovator product compared to the strain 1 product seen at the bottom. If you take a look here at the figure on the right, you see the profile of the strain 2 and innovator materials tested after treatment with sialylase A. And you can see that the complex profiles that we see on the right are vastly simplified after treatment with the enzyme. And you'll notice that these 2 proteins have almost identical charge profiles after digestion, which suggests that they both likely contain similar protein structures without their sialic acid. Okay. And I would just like to wrap up by saying I hope I provided a few examples of how image capillary isoelectric focusing can be particularly useful in vaccine and biopharmaceutical process development. I'd just like to acknowledge a few people. In particular, I'd like to acknowledge my manager at the time, Richard Rustandi, and I'd also like to acknowledge Brian Piklansky for generating most of the data shown for the recombinant protein. Thank you. Back to you, John.
Thank you, Carrie, for that excellent demonstration on the use of ICIEF in a range of applications. I think your presentation very nicely complemented Scott's talk in terms of showing the advantages of ICIEF in a biopharma environment. I'd like to remind our audience that we will be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist is Karan Shah. Karan, the floor is yours.
All right. Thank you, John. And hello, everyone joining us today. I'm going to be talking about analysis of maytansinoid ADCs, that is antibody-drug conjugates by imaged capillary isoelectric focusing. I will begin my talk with a brief introduction to antibody-drug conjugates made with Immunogen's maytansinoid ADC technology, and then move on to talk about analytical methods that have been traditionally employed to monitor charge heterogeneity, and then talk about ICIEF in general for antibody and ADCs and the advantages that it offers over the other techniques, and then some method development work for ADCs, and then lastly, I will talk about an application of ICIEF, which is very useful in quantifying the levels of unconjugated antibody in ADC samples. So to begin with the introduction, maytansinoid ADCs, they utilize high specificity of an antibody with the cell-killing ability of a small molecule cytotoxic agent. There are three main components to the ADC molecule. The antibody itself, which is the tumor antigen specific, and all antibodies that are being used in clinic right now with ADCs are either IgG1 or IgG4 type. The second part of the ADC molecule is the linker molecule, which is a bifunctional linker and attaches to the antibody via the lysine residues. And it has the cytotoxic agent attached to it on the other end. And this linker, by its chemistry, can be reducible or non-reducible, and sometimes also referred to as cleavable or non-cleavable. And the last part of the ADC molecule is the small molecule cytotoxic agent, which in this case is a maytansinoid type of a compound. Maytansinoids are members of the ansamitosin family, and they're highly potent with their IC50s in the range of picomolars. And the mechanism of action is that they kill the cancer cells by tubulin polymerization. So this slide, we basically look at the basic structure of a maytansinoid ADC molecule, wherein we see that the different maytansinoid drugs that are attached onto the antibody via the help of a linker. And on the left, you have the example of the small molecule cytotoxic agents, which are DM1 and DM4. And on the right, we have the examples of the linker molecules, like the SPP and SPDB, which are the reducible type of linkers, and then the SMCC, which is a non-reducible type. Typically, all ADC molecules would have 0 to 8 maytansinoids attached to the antibody, and on an average, the drug-to-antibody ratio is about 3 to 4. And the linker choice is dependent on the antibody, the target that it's made for, and also depends on the indication for that particular conjugate. So this slide shows the basic mechanism of work of how the ADC compounds work. And once inside the bloodstream, the ADC molecule identifies the cancer cells via specific receptors and binds to the cells. And then once it is internalized by its natural processes, the small molecule is kind of released from the ADC and is free to act on the cancer cell and kill the cancer cell. So this is a very elegant mechanism, like the magic bullet. It's very targeted therapy. Now moving on to the charge heterogeneity in ADCs and why it's important to look at them. To begin with, we know that the antibody itself can contain a lot of charge heterogeneity due to the C-terminal lysine, the N-terminal pyroglue, deamidation, and other modifications. And then when the antibody is made into a conjugate by attaching linkers and small molecules to it, the lysine, being a basic amino acid, the PI of the antibody decreases each time that an amino group of the lysine is modified by a linker. So ADCs in general, they comprise like a mixture of these molecules where each antibody molecule has different numbers of DM1 and DM4 covalently linked to them. And that is why the charge heterogeneity is complicated and is very important to monitor to understand and characterize ADCs, and also to look at the consistency of manufacturing from lot to lot and batch to batch. Now, traditionally, we know that ion exchange, slab gel, IEF, and capillary isoelectric focusing have been used to look at charge heterogeneity in proteins, but each of these come with some of their limitations when you compare them to ICIEF. And the main ones being, in general, the resolution is poor. There's always difficulty in quantifying the protein, and some of these methods are also very time-consuming and labor-intensive. And I will go through some examples of these methods. So here we have the analysis by weak cation exchange for an antibody and its corresponding ADC. We can see that the cation exchange method works very well for the antibody, but when you go to the antibody-drug conjugate, see there is hardly any separation of the charged species, and it just comes out as a big, huge peak, and you can see a small amount of unconjugated antibody in it. Going on to the next example, you have the separation of an antibody-drug conjugate by a traditional ICIEF method. And although the resolution is slightly better than cation exchange, it is nowhere close to what you can attain by ICIEF. And you still see that the peaks are kind of separated, but it's still very noisy and unclear. So ICIEF is done using an ICE280 and ICE3 system, and it has a very high throughput compared to the other CIEF methods. And from what experience that we have had with ICIEF, it has very high PI reproducibility and a high resolution of charged-based species. And the good part about the data is that these electropherograms are easily converted, and you can export them for integration and quantification purposes as well. So this one just talks about basic sample preparation and ICIEF analysis, and a lot of this was covered by Scott in the beginning, so I'm not going to go into much detail, but it just involves like a sample prep wherein you add your analyte into a proper amount of methylcellulose, ampholytes, and PI markers, and then other additives. And then analysis is done on the fluorocarbon-coated capillary with analyte and catholyte solutions. And then the detection is done using a CCD camera at 280 nanometers wavelength. So this basically shows the electropherograms of an antibody and its corresponding ADC by imaged capillary isoelectric focusing. So here we see that on the top figure, you have the antibody main peak, and then you have its acidic and basic variants, and they have a very well-defined PI. And when this antibody is conjugated with the small molecules and linkers, what happens is that each time the lysine is modified, the PI of the antibody decreases, and so you get this whole distribution of various charge species towards the left, and each of them indicate that an additional drug molecule has been attached to the antibody. And when we look at the PI of the main peak of the antibody and the PI of the rightmost peak on the ADC profile, they correspond very well. And we have verified by various spiking studies that the first peak, the rightmost peak in the ADC profile, actually is the unconjugated antibody. So it is very useful, ICIEF, in monitoring the charge heterogeneity of antibody and antibody-drug conjugate samples. Here we compare the ICIEF profile generated for an antibody-drug conjugate to a mass spec profile. And we see very well that the charge distribution profile that both these methods give out are very comparable. It's just that because of the different mechanism nature of these two methods, the profile distribution is in the opposite order. So where for the ICIEF on the top, you have the unconjugated antibody peak on the rightmost side, which is the P1, and then the different conjugated species, they span left. For the mass spec profile, you have the unconjugated antibody coming out on the left side, and then as drug molecules are attached to the antibody, its mass increases, and you have this mass distribution profile spanning right. But this also shows very well that ICIEF is capable of generating data which is very comparable to what we get from sophisticated methods like mass spectrometry. And then I will talk about some method development of ICIEF for ADCs and some of the examples of the parameters that affect ICIEF separation are focusing time, the ampholyte composition, additives in the sample prep like urea, and sometimes even interferences from formulation buffer components of your analyte itself. And I will talk about some of these examples in details. So to begin with, this slide shows the analysis of a particular antibody-drug conjugate using two different ampholytes. On the top, you have the servolite, and on the bottom, you have the ADC profile using a pharmalite. And due to the high PI of this antibody, the choice of the ampholyte was anyways limited. And here we tested servolites and pharmalites. And as we can see clearly from the figures, that servolite for this conjugate did not work very well, and you don't see very clear separation of species, and there is some noise in the baseline. But with using pharmalites, we have a very clean and nice baseline, and nice resolution of many species of the ADCs is seen. So carrier amphelite composition is one of the considerations that you need to look at when you're doing method development. This one here we see how sometimes you may require additives in your sample prep. Here we have an example of a conjugate that was analyzed without having any urea in the sample prep. And you see in the electropherogram that there's a lot of precipitation and noise in the profile, and there's hardly any charged base separation. But when we added some urea to the sample, it helped keep the sample soluble while it was being focused. And you see a very nice and clear separation of charged base species, and the resolution is fantastic. So addition of some of these additives might be necessary depending on the nature of your analyte molecule. And lastly, for method development, we have an example wherein a component of the formulation buffer was interfering with the analysis. And here you have the same ADC molecule that was analyzed in two different formulations. On the top, we have in an acetate formulation, and we see there's a very nice separation of charge-based species. But in the bottom figure, as you see, this was the ADC sample in a histidine formulation. And histidine being an amino acid has a pI of 7.6, which kind of overlaps with the range of the pI of the ADC molecule. And we see it focuses at that point, and it displaces the charged species of the conjugate and creates this gap. And that's simply because the histidine does not absorb at 280, and you see just a plain gap in the profile. And this issue can be easily resolved by doing a simple buffer exchange of your analyte into a compatible buffer. So sometimes even your formulation buffer components can interfere with the analysis and need to be considered for method development. Coming to the last part of my talk, I will talk about analysis of the antibody by ICIEF method and how this method was evaluated for various parameters like reproducibility, linearity, precision, and then eventually this method was used to look at the level of unconjugated antibody in our conjugate samples. So this slide, here we see on the left top figure, we have the antibody calibration curve, wherein the different levels of the antibody were put and analyzed, and you see the electropherograms in an overlap. And at the bottom in the graph, you see the linearity, and it was found that this antibody calibration curve was linear in the range of 0.001 to 0.1 milligrams per mil. And even the reproducibility of the PI for various injections, the RSD was almost like 0.4%. And the LOQ of the method was found to be around 0.002 mg per mil. And even when the analysis was done on multiple occasions, there was no change in the PI. And the day-to-day precision and recoveries, they also looked great, less than 20% RSDs. And for concentrations greater than 4 micrograms per mil, the recoveries were all well within 80 to 120%. So we used this data, and then we generated a calibration curve of the antibody, and then we analyzed the conjugate sample and tried to quantitate the unconjugated antibody in the conjugate sample. This slide talks about two different approaches that was used to look at the UMAB level in the ADC. The first one was just by a function of total peak area, and the second one was actually using the calibration curve as I talked about in the previous slide. And it was found out that calculating the percent UMAB as just a function of total area is inaccurate, and that is because of the contribution of the absorbance at 280 nanometers by DMX molecules. So what happens is in the conjugate species, as the antibody becomes conjugated, you have this DMX, the small molecules attached to the antibody, which start to contribute to the absorbance at 280. So you have a kind of overestimation of the highly conjugated species. And this degree of overestimation is dependent on how highly conjugated that particular molecule is. So ICIEF tends to slightly overestimate the higher conjugated species. And as a result, you see by the first approach, the percent UMAP calculated were all slightly less, as in the top line in the table. And when we used the calibration curve, and we found out from orthogonal methods as well, that these numbers were more accurate and more consistent with what the UMAP levels in the conjugate. And also the level of the unconjugated antibody in ADC samples was higher than the LOQ, so you could rely on the method for accurate estimations of the UMAP levels. And this slide basically just shows a similar example for another ADC molecule. And here you see in the figure in the background the antibody calibration curve of different concentrations. And it's very linear response with R squares greater than 0.99. And on the top trace in the green, you see the conjugate and how the first peak in the conjugate corresponds very well to the antibody peak. And the unconjugated antibody level was estimated from all this data. And it was found to be very comparable when it was looked at by other orthogonal methods like NHPLC or mass spec. So ICIEF provides really useful tool in quantifying unconjugated antibody in conjugate samples. And lastly, just going through the whole presentation, we looked at charge heterogeneity in antibodies and ADCs and how it can be monitored by ICIEF with very high resolution. We looked at how ampholyte selection, focusing time, additives, and formulation components need to be considered for method development. And the charge profile from ADC that is generated using ICIEF is very comparable to that obtained by mass spectrometry. And then also, lastly, the application of ICIEF, where it is very useful to look at the unconjugated antibody levels in an ADC sample. And this is the data shown here was all for the methods that have been working, but we do have antibody-drug conjugates wherein we cannot develop for unknown reasons a proper working method by ICIEF, and then we have to look at other methods. But for conjugates which have a working method, all these things are very useful, and ICIEF provides a very useful tool in looking at charge heterogeneity and other parts of the conjugate. That's it for my talk. I have a few acknowledgments. I would like to thank everyone in the analytical department here at Immunogen, and Alex, Godfrey, and Mike for their support, and Joyce and Amy who generated data that was used in the presentation. That's it for me. Back to you, John.
Thank you, Karan, and thank you, Scott and Carrie, for your description and insights on the use of ICIEF in various applications. I think our audience definitely appreciated your talks because they've sent in some very pertinent questions. We're almost ready to begin our Q&A session, but I'd first like to ask you to disable your pop-up blockers because a short survey on this webinar will be appearing in a moment. We would very much appreciate your feedback on this webinar. Thank you. Okay, so we're ready to begin our Q&A session, and let's take the first question from the audience.
Okay, Scott, we have the first question for you. Do you have robustness study for HT cartridge? I guess a robustness study for an HT cartridge. How many injections can be injected through the HT cartridge before it expires or retires?
Thank you very much, John. Actually, we have extensive verification and validation data on the new coating type for the HT cartridge. We guarantee at least 100 runs on the HT cartridge. With that said, one thing that must be taken into consideration, since it is a hydrophilic coating, that having the capillary stored with catholyte can damage that coating. So we stipulate that you should minimize exposure of that capillary to the catholyte idle without the system running for less than 4 hours. Hopefully that answers that question.
Okay. And Scott, do you have any recommendations for an ampholyte composition of a monoclonal antibody with a PI of approximately 9.6?
Yes, you can typically use a 4% 3 to 10 gradient and then use a 5 millimolar arginine cathodic spacer to initially screen for the compound. Typically, I'll use between 2 and 4 molar urea to make sure that I don't get any aggregation or precipitation events. If resolution isn't sufficient, you can then add in 8 to 10.5 Pharmalite, and I typically start at about 2%, pulling back my 3 to 10 Pharmalite from 4% back to 2%, so I have kind of a equal volume to weight ratio for the amphiolites.
And Scott, what is the highest PI that can be used with ice and still have a good resolution, reproducibility, et cetera, et cetera?
Typically, the limitation is usually the cathodic blocker. So the isoelectric point for arginine is somewhere around the range of 10.7. So you can actually use the 8 to 10.5 to get good resolution out to about 10.5. If further is needed, you can use the 9 to 11 Servolite with TMED. And typically what I'll do is that I will use about 2% or less of the Servolite just due to the background noise, and then between 5 and 10 millimolar of the TMED.
And Scott, someone may have missed part of the webinar. He says, maybe I missed it, but what is the minimum protein concentration required? Also, what kinds of excipient interference do we need to watch out for example, between salts, et cetera?
The most important thing is that to maintain a ionic content less than 15 millimolar PBS. Actually, there is a significant study that was performed by Jiaji Wu on the subject for minimizing ionic components in the ice and also CIEF systems. That what those ions do is that they compress the gradient, because as they leave the capillary, they're replaced with hydronium and hydroxyls, which cause extreme pHs at the end of your capillary. Other excipients, obviously things like Triton X100, things that absorb in the 280 range are not going to be very beneficial for sensitivity. And looking at concentration, this is the hard thing about isoelectric focusing. Unlike, say, CSDS, where you get a single peak, you get actually, you can get a wide range of charge variance. So that can cause a dilution effect. So if you have, same concentration of a compound with the same molar absorptivity, the concentration per peak would be, say, twice as much if you only had 2 peaks as opposed to 4 peaks if all peaks were equal in area. Typically where I start off at is that I start off at 200 micrograms per mil of a monoclonal antibody. You can go down as far as 50 micrograms per mil and still get relatively good detection if the compound has a low enough charge heterogeneity.
Thank you, Scott. And does imaged CE offer any advantages over CIEF, the ICIEF method from Beckman?
Yeah, that's kind of asking, like with me, that's kind of asking about my child being ugly. I have experience with both methods. I did do most of the development for the Beckman method while I was at Beckman. That the biggest advantage that you run into is the elimination of the mobilization step. That really, it significantly truncates the analysis, which allows for much higher throughput. And it also does eliminate the effects of chemical mobilization. So with the Beckman method, that you actually have a 2-step separation, that you have isoelectric focusing on the bottom end, and then on the top end is a chemical mobilization, which is actually more of a free zone. So you can get effects based upon differences in the mobility of the compounds. And that's why we see the expansion of really that 8 to 9 range, which causes PI shift effects when we see the chemically mobilized gradient as opposed to the static imaged ICF. With that said, that really the big advantages that you run into with the ICE method are the speed and the fact that you're not really affecting the gradient during the detection step.
Thank you very much, Scott. Carrie, a question for you. What kind of autosampler has been used in this study for quantifying concentration?
We've actually used two different autosamplers. So one was the system with the Prince autosampler, and we also had another system that we used which had a 96-well plate. So we actually were able to use both systems, and in large part depended on the number of samples we were receiving.
Okay, and another Carrie. Regarding protein interaction and PI shift, is the assay under denaturing conditions, that is, containing urea?
No. In this case, we did have glycerol in there. We noticed when we did some development that we needed to include that as an additive for the most reproducible profile, but we do not include urea in the sample prep for that protein.
Thank you. And Karan, can you comment on the requirement for urea on separation?
Sure. The thing is, when we are dealing with antibody-drug conjugates, they are slightly more hydrophobic than just antibodies due to the attachment of the small molecules. So it is a challenge to keep them solubilized while they are focusing. And so what urea does is acts as a mild denaturant and kind of slightly opens up the conjugate molecule and exposes the charge of the conjugate better, which helps to separate based on charge, and you see this good resolution. So that is why we found urea to be really helpful, especially in cases of conjugates. For antibodies, we typically see that urea is not required as such, but its addition does help in giving you a good reproducibility from time to time, and you don't see these injections which have these precipitation and spiking issues.
And Karan, how much urea did you use for your sample prep?
So for the ones that I showed in this presentation, the urea that was used was around 4 molar in the sample prep. But typically we check anything from 1 molar to 4 molar, and we have not yet gone above 4 molar urea in the sample prep. That is kind of the upper point that we've tested. So I would say around 2 molar urea should be good enough for stabilizing effect.
Okay, and Karan, have you ever seen that using urea while improving protein precipitation issues also decreases your resolution?
That is a good question. We do have products which do not require urea to keep them soluble while they are focusing. So when we look at the electropherograms of these products with and without urea, we see that there is virtually no change in the resolution. So I would say that we haven't seen any of the bad effects of urea on resolution, and it only kind of seems to improve it and keep it more reproducible. So also a thing of caution is to, when we use this urea, it's always freshly made, a freshly made urea stock, and maybe if you are using old urea, that can kind of have a negative impact on your sample. And that's why we recommend using freshly made urea stock solutions.
Thank you, Karan. And Carrie, question for you. For the protein concentration determination, the calibration curve is based on the total peak area. Is the calibration curve based on the total peak area or main peak area? Any sample pretreatment? Carrie, did we lose you?
Hi, I apologize. Sorry.
Okay. All right.
Technical glitch.
Okay, let me read that again. So for the protein concentration determination, is the calibration curve based on the total peak area or main peak area? Any sample pretreatment?
In this particular case, we actually used the main peak area. So I showed here a concentration for our standards ranging from 500 to 63 microgram per mil. Later on in development, we actually looked at extending the curve range down to 15 microgram per mil. So on the lower end of the curve, you don't actually see the acidic variance. And again, for this particular protein, the acidic variants are a small portion. So we figured between the fact we had to look at some dirty samples where we probably wouldn't be able to differentiate the acidic peaks from other proteins that might be present in those particular sample types, and that we would not see the acidic variance on the low end, we decided to use the main peak area only to be consistent as possible. And there was no sample pretreatment. In this case, we just diluted the sample right into the amphylate mixture and tested it. So I hope that answered the question.
Thank you, Carrie. And Karan, given the similar ADC subspecies distribution profiles you observed for CIEF and mass spec, have you tried to use CIEF to determine the drug-to-antibody ratio on your ADC?
Yes, we actually have used the CIEF profiles to determine the drug-to-antibody ratio. And as I discussed in the presentation, what happens in ICE is that even the small molecule on the ADC contributes to the absorbance at 280. So what we are doing is we are slightly overestimating the higher conjugated species. So when you look at the drug-to-antibody ratios calculated based on CIEF, those tend to be slightly off compared to what you get from mass spec, but they are still very comparable, I would say. So yes, we have used it, and those slightly off, they give you a good idea about the drug-to-antibody ratio.
Thank you so much, Karen. Well, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or, and we highly recommend this, recommend it to your colleagues and friends. Thanks again to our panel for the outstanding presentations, and I want to say thank you to our audience for your attention and for your very thoughtful questions about a number of topics that were brought up during the webinar. And thank you to our sponsor, ProteinSimple, who made this event possible. Bye for now.